You're closer to some of our students. Yes, Douglas Cormay is a researcher at the University of Arizona in Tucson, USA, and he's the director of the cellular imaging facility, amongst other heavy responsibilities. So he's going to talk to us today about ethics in imaging. And please, Douglas.
All right, let me share my screen. And all right, well, we were laughing before you all came on about whether this is morning or afternoon. It's morning for me. Uh, this picture, I was looking at it as I was getting ready this morning, going, is the sun coming up or the sun going down? I'm not sure. Um, but thank you for having me, Sylvie. Let's get started. So when we work in science, there are a lot of things that are regulated. If you do human subjects research, if you work with research animals, biosafety, radiation, lasers, chemical safety, all those things are regulated. And, violating government regulations can result in fines or even sometimes criminal prosecution. In the US, we have two major government granting agencies, the National Science Foundation and the National Institutes of Health, and they're very concerned about misconduct, and they use a similar definition. And it really comes down to these 3 points, the fabrication, falsification, and plagiarism. And you'll encounter these if you do work with the US at all, but in many other countries, they are also uh, important concepts about how to work with research data. And one thing that I always point out to people when we talk about this is that there is this little caveat that says that research misconduct does not constitute or does not include honest error, which is sometimes hard to prove that errors were done honestly, um, or differences of opinion, and we'll come back to that in a moment. And like I said, Sweden has similar language, but they talk also about deviations from good research practice and negligence, which is, negligence, sloppy science. So the first two, the fabrication and falsification, are where a lot of problems happen, but there are also issues with people stealing images from other researchers and reusing them. And it's taken, let me move this out of the way here. It's taken quite a while for science to come to an understanding about what
Yes.
actually
Oops,
constitutes.
sorry. I think you were frozen for a little while, like for you stopped after basically your first sentence there, and then you were frozen. Maybe you can repeat uh, what you were saying.
My first sentence on this slide.
Yeah, we heard that one, but then afterwards that was gone.
Okay,
Thank
so
you.
sure. So the idea of good scientific practice with digital images is still a work in progress because what's appropriate in the electron microscopy or the atomic force world is not necessarily appropriate for fluorescence microscopy. So it makes it hard for scientists to agree on what's the best way to work with digital images. And I really try to emphasize to my users that this idea of the differences of scientific opinion do not constitute misconduct because the bottom line is if you tell people exactly how you manipulated an image, your protocol for that, then a reviewer or a reader might consider that you don't have the skill to properly do that, but that you're not cheating. And I said something about this at a meeting 2 years ago. It happened to be with somebody from the Office of Research Integrity, which investigates misconduct in the United States. And I watched the man not bob his head up and down. So I think I'm onto something. So let's see how bad the problem is. Elizabeth Bick, who you may have heard about, is someone who's got a real eye for looking at image manipulations. She looked at over 1,000 papers, primarily looking at gel images, and she found a little under 4% had suggested manipulations, and with 2% of them looking very suspicious. The Office of Research Integrity, I looked up their cases in this time period, and there were about 80% of them had uh, misappropriated or misused images, including a case from someone from the University of Arizona who I knew. And I looked at the Retraction Watch database of um, retracted papers and found that about 14% of those included the words related to image manipulations. So it's a problem. And just to give you an idea of the scope of this, some really highly powered investigators in the US were made the news in the wrong way. The Dana-Farber Cancer Institute is associated with Harvard. They had multiple papers from multiple investigators that were cited. And this faculty member, Dr. Maslia, was an advisor to one of the institutes within the NIH and was a very well-known Alzheimer's researcher. And it's just bad news when this kind of stuff gets in the papers. But we don't want to hide it. We need to fix it. This is what we need to do. So thinking about what you've already learned, let's look at a couple of things that you might want to consider when you're dealing with images. The first is about your optics. Now, I love those images that you see in the contests, the Nikon small image contest, Olympus has a contest, others have contests. They're fabulous images. And if you look at most photography contests, if you're competing against professional photographers, they have nicer gear than we do. I take pictures with my cell phone. I don't have a single lens reflex camera. But the people that win the microscopy contests are often using the same kind of equipment that's found in your core facilities. So what's the difference? Well, I've often said interesting looking samples help, but the thing that really separates the winners from everyone else is that they are meticulous and careful about their sample preparation. And that's the big differentiator between junky kind of images and beautiful ones. The, because the sample is part of the optics of the microscope. And a colleague of mine has said that 90% of good imaging happens at the bench. So some other things you probably talked about detectors, so I won't go into this in great detail, but there are issues with bit depth, about increasing or decreasing that, sampling, noise in the image, either from the detector or we just have a lot of background, uh, the issues of saturation. There's some beautiful pictures in the lobby of our cancer center here that were taken by a faculty member friend. They're all saturated. That they're nice art, but they're not really good science. And what can your histogram tell you about your image? Learn to look at that and pick up some things about what's going on in the image. With a digital image, it's part of the analog to digital process of getting from what you see by eye and what becomes part of the digital image in the detector. And it's not just a picture, it's data. And it has some limitations. And it's a numerical sampling of that analog data. It has spatial and temporal, spectral characteristics, and other dimensions like different wavelengths, locations, focus stacks, and they're all what we call bitmaps or maps of intensity. So if you take an image and you turn it into an Excel spreadsheet, you end up with numbers that are registered one to another and different intensities. And it comes in a container. It's a file format. Often they're hard to open if you don't have the right proprietary software. or you don't have the bioformats plug-in, and they have typically, especially on the more automated microscopes, a considerable amount of metadata related to how the image was acquired. If you export your image to TIFF or other common file formats, there's often a loss of that metadata. And I just take a moment just to say that JPEG is not a recommended file format for scientific images. Just don't use it. We could go into why, but it is not really the time for that. So just remember, when we go from proprietary file formats to these other formats, that's more convenient because we can work with them in Photoshop or GIMP or whatever you work on your images with, but you lose a lot of metadata in the process. And really, all the functions that are applied to a digital image are mathematical. You have that spreadsheet of numbers, and you are applying some sort of function to it to get the pixels to look differently. And it's often impossible to detect what kind of post-processing that has been done. You know, people are good at seeing some things in images, but other things are very hard to tell. And things like a histogram stretch and other manipulations are considered fairly benign, but you want to be reporting as much information about how you process the image as possible. And sadly, a lot of people treat the difference between what they get and what they present. The stuff that happens in the middle is just sort of a black box. They don't know. And that can cause some problems as well. Um, Vendors will often overpromise what their software can do or is doing and don't always explain it well. So we're going to work through the discussion questions. Um, as I understand it, Sylvie's assigned a question to each group, and then there is the final question that we're all supposed to work through and come back at the end, and we'll talk about what you've learned and what you've come up to, and I'll let Sylvie.
Yes, exactly. So, thank you, Doug, for this nice introduction. And we have now a group discussion. So we're going to go into the other Zoom room. And Doug, I think I have sent you the link, but if not, I can send it to you now in the Zoom chat. Okay, so I see you all in the other Zoom.